Good evening, everyone, and a very warm welcome to our distinguished speakers, respected doctors, researchers, and laboratory professionals and faculty members, students, industry experts, and participants joining us from across India and beyond the globe. It is truly an honor and a privilege to welcome you to the inaugural flagship webinar of UDGAM AI and Health Research Center, Healthcare 2030, where digital health, AI, diagnostics, and research converge. Today is much more than the launch of a webinar. Today marks the beginning of a vision where healthcare is not limited by traditional boundaries, a vision where healthcare is not limited by the traditional boundaries and clinicians collaborate seamlessly with engineers, biologists, bioinformaticians, data scientists, and artificial intelligence specialists. A vision where cutting-edge technology serves one purpose alone, to improve human health. And it has always evolved through innovation. Discovery of drug transformed medicine. Medical imaging revolutionized diagnosis. Genomics changed our understanding of disease. And today, artificial intelligence is ushering in the next great transformation. And AI is no longer a futuristic concept. It is already assisting radiologists, pathologists in interpreting images, helping pathologists analyze digital slide, accelerating drug discovery, supporting clinical decision making, and enabling precision medicine. Yet technology alone cannot transform healthcare. The future belongs to collaboration and validation. And it belongs to institutions willing to embrace innovation while upholding ethics, compassion, and scientific integrity. This is precisely why UDGAM was created. And before we begin today's scientific session, allow me to introduce the organization behind this initiative. UDGAM stands for Unifying Diagnostics, Genomics, Analytics, and Medicine. The name itself reflects our philosophy. Healthcare challenges are becoming increasingly complex. No single discipline can solve them alone. The future of medicine lies in bringing together diagnostics, genomics, artificial intelligence, computational biology, laboratory medicine, clinical sciences, engineering, and translational research facilities into one integrated ecosystem. And this is exactly what UDGAM represents, a platform where medicine meets technology, where research meets clinical practice, where innovation meets responsibility, where education meets real-world implementation. Our mission is simple but very ambitious: to build an interdisciplinary ecosystem that empowers and bolsters healthcare professionals with the knowledge, tools, and technologies needed for the future of medicine. Our vision is to prepare the next generation of healthcare leaders capable of working confidently with artificial intelligence, omics-based technologies, digital pathology, computational diagnostics, bioinformatics, precision medicine, and health translational research. Our guiding principle is reflected in our tagline, that is, innovation intelligence for better health. Through scientific webinars, certification programs, workshops, and collaborative research initiatives and partnerships with academia, hospital, diagnostic centers, laboratory, and industry, UDGAM aims to become a catalyst for innovation in healthcare. Whether you are a clinician, researcher, or laboratory professional, pathologist, biotechnology student, engineer, bioinformatician, or AI enthusiast, UDGAM welcomes you all to become part of this growing community. And this webinar is our very first step. And it certainly will not be our last. It would be just an initiation. And we hope today's discussion inspire collaboration that continue well beyond this event. Over the next 1 hour, we will hear from distinguished experts working at the forefront of artificial intelligence, digital pathology, molecular diagnostics, and oncology. And today's discussion will explore how AI is reshaping the healthcare, not as a replacement for clinicians, but as a powerful tool that enhances diagnostics, treatment planning, and patient care. And for following each session, we will have a short interactive discussion and we'll make an audience dais for open question. So now I am just going to introduce our first keynote speaker, Dr. Utso Guha. Dr. Utso Guha is a distinguished consultant pathologist and AI in healthcare specialist with over a decade of clinical experience working at the intersection of laboratory medicine, digital pathology, and responsible clinical AI adoption. His educational journey reflects an exceptional commitment to integrating medicine with emerging technologies. He has completed his postdoctoral certification course in digital pathology. The certificate course is in artificial intelligence in medicine and holds ABAIM certification from the American Board of Artificial Intelligence in Medicine. In addition, he has received professional training in AI management system, highlighting his experience in responsible AI governance. Dr. Roy has served as faculty and invited speaker in numerous national healthcare and AI programs, delivering lecture on AI-powered laboratory medicine, digital pathology, breast biomarker reporting, clinical decision support system, health data privacy, and ethical AI implementation. His work focuses on translating artificial intelligence into practical, clinically meaningful, and ethically responsible solutions that improve diagnostics accuracy and patient care. He is active member of several prestigious professional organizations, including the Indian Association of Pathologists and Microbiologists, the Digital Pathology Association, the Asian Society of Digital Pathology, the Indian College of Artificial Intelligence in Medicine, and the Calcutta Association of Practicing Pathology. Today, Dr. Roy will share his vision on how artificial intelligence extends far beyond conversational AI and is helping build the intelligent healthcare ecosystem of the future. Thank you, Dr. Roy. Now the dais is open for the discussion and your insightful lecture. Thank you.
Good evening, everyone. Hopefully, I'm audible. Yeah. Okay, so I will just share my screen. So, good evening, everyone, and warm welcome.
Sir, you are not audible. Please turn on your microphone.
Yeah, am I audible?
Yes.
Okay, fine. Thank you. So, yeah, good evening, everyone, and a warm welcome. So, a very warm welcome to first venture of UDGAM. I will just share my screen. Okay, I hope the screen is visible.
Yes, sir, it is finally visible now.
Okay, great. So, as Ramomita was saying, so it's our first live webinar. So first topic of my topic is actually beyond ChatGPT and building an intelligent healthcare ecosystem of 2030. So it will be by 2030, the healthcare won't be run by one clever model. It will be most likely run by a system, like a mind. So built from many intelligences working together. And that's what I want to show you over the next 15-20 minutes. So we are saying as beyond ChatGPT because the future of healthcare isn't a better chatbot. It's a comprehensive, intelligent ecosystem. And let me explain why that matters very importantly. So the question everyone in healthcare is asking sometimes is how do we make this chatbot better or that chatbot better? How do we get smarter answers, fewer hallucinations, nicer bedside manners? So that is kind of a misguided question. So the real question is how we can build the intelligent frameworks that integrate many components into real clinical workflow. And one feature is the first one is a feature request. The other one is more of a system design problem. So a chatbot, what we understand, actually responds to a prompt. That's all it does. It has no memory of a patient an hour ago, no ability to act upon a lab value, no way to close the loop. An intelligent healthcare system is different. It doesn't just respond, it acts on information, processes the data continuously, and closes the feedback loop around the patient. That gap between responding and acting is the entire story. So the shift is from a single model to compound systems. So healthcare AI is moving from one model answering the one question to an orchestrated system, specialized system of agents or agentic AI and tools working in concert with the clinician's judgment still firmly in the loop. Notice, I didn't say just replacing the clinician. I said human oversight remains crucial. And hold on to that line because that is the thread that should run through the whole talk and through our own coming days also. So generative AI itself will stop being a novelty like it is now and become actually an infrastructure like electricity, not like a magic trick. It will quietly power the applications that read the data streams, flag the draft anomalies, drafts reports, and make workflows more efficient, mostly invisible to the patient. And that's the maturity curve every transformative architecture, every transformative technology must follow. And healthcare AI is now on that same verge. So if a chatbot is just a small part, what does the whole system actually look like? Let's explore it layer by layer. This is an intelligent healthcare stack. So this is my central diagram for tonight. So I will linger on it for a few seconds. So it has actually 5 layers. Obviously the data layer, then there will be models, then there will be orchestration layer, followed by the workflow layer, and on the top of it will be the human oversight or the oversight. No single layer is actually the AI. The overall intelligence lives in how these layers talk to each other and improve the overall healthcare and the patient outcomes. So coming to the layer 1, that is the data, it should be interoperable and it should be standardized for all healthcare data. So we have ABDM in India, FHIR in globally, so that information can move safely and instantly across platform. So you have done a blood test in Kolkata, you can access it at Delhi also when you are shifting to a different hospital. So without this layer, nothing above it or below it works because you cannot orchestrate what you cannot connect or what you cannot analyze or view. So we have to have the data accessible and interoperable. Next comes the model layer. So here where the foundational models will meet the task-specific diagnostic models. For example, a general reasoning model can be paired with a purposefully built model for, say, radiology or cytology or ECG interpretation. So that will give you both context, breadth, along with a precision. So both will be combined and not competing with each other. Layer 3 will obviously be the orchestration. So this is the coordination layer where multiple agents will be reasoning, calling tools, and handing off tasks to one another and crafting one integrated flow that instead of a dozen of disconnected apps or disconnected softwares. So this is the layer that makes that beyond ChatGPT actually literal, the agents that can reason, that can act, not just model that only replies to one specific question. layer flow will be the workflow. So all of this is meaningless if the software or the AI will live in a research demo or maybe in one computer in one corner of our lab or one of our hospital. So the workflow layer should embed these AI solutions into real clinical lab or lab pathways. So that they should have been integrated into the actual order sets, actual reporting screens, so that the technology disappears seamlessly into practice rather than sitting beside it where nobody is using it because it adds an extra step to our workflow. And the layer 5 is obviously the one that I will also come back at end, that is oversight or human validation and accountability. It runs through every layer below, and this is what earns the trust of the automated decisions. Keep this layer in mind so that I can talk about it again in the later. So here's why the architecture matters practically, because every powerful diagnostic tools that will come in the coming days, maybe digital pathology, maybe proteomics, maybe transcriptomics. So all will be like a module, a plugin. So it only delivers a real value if it sits inside a cohesive governed system like the one I just showed you. So any brilliant components without a system around them will just stuck in the pilot studies or maybe a very well popular research publication. So to see a clinical implementation, it has to enter a well cohesive system. So now that you have that system, something remarkable starts happening. So it's how we discover medical knowledge in the first place. So the discovery cycle itself is evolving at a rapid pace now. So we have recently the Nature publication of Google's AI co-scientist, where it has shown that a multi-agent system can repurpose drugs for AML also. And in one case, we derived a resistant mechanism in about 2 days that actually had taken the human researchers team around 10 years previously. I want that number to sit on your mind for 1 second. That is 10 years compressed to only 2 days. So this was published just in the May in Nature. Then there are autonomous research agents, systems that don't search the literature, they help conduct the experiments, analyze the data, draw conclusions with real independence, and dramatically expedite the pace of the discovery. Think of it as giving every research team an extra tirelessly fast collaborator. At the molecular level, the biological foundational models like AlphaFold 3 that won actually the 2024 Nobel Prize in Chemistry can now predict protein structures and cellular responses computationally. So that means that understanding disease mechanisms and screening potential future therapies becomes easier even without doing a single wet lab experiment initially. So which has paper showed recently that 3.3 million scientists in more than 190 countries are using Google DeepMind and AlphaFold from their database for their research activities. And the experiments themselves are starting to run without waiting for us. Self-driving labs pairs robotics with machine learning to automate experimentation. USA's Berkeley's A Lab is a landmark example. So it closes the loop from hypothesis to synthesis to results and accelerating the path to practical application. I will try to run a video if it's possible. It's from their lab only. It shows how an automated robotics system is doing an experimentation. It's mostly for metallurgy, I think. So it's mixing the reagents, all titration, everything is done robotically. So the results, obviously, we will see will be much better than a human doing by hand. So I'll come back to my presentation. But none of this will work if the hospitals cannot share the data. So federated system solves that. It is a system where it trains models across multiple hospitals without the data, patient data actually leaving the building. So the exam, that is the EMR chest X-ray study done during the COVID days, one of the best examples of this, where a federated model outperformed individual single site efforts while fully safeguarding the patient confidentiality. And that is exactly where the global south, that is our countries and others, stops being an afterthought and becomes central role play. So federated learning lets regions like ours contribute meaningfully to global healthcare, even under strict data protection law, without ever exporting a single patient record. India isn't waiting to receive this technology. We have the digital public infrastructure like ABTM to help shape it. And which brings me to the last and in some ways the most important layer of tonight's talk is governance. So effective governance isn't a brake on the innovation, it's the substrate that makes innovation trustworthy enough. Validation, accountability, and regulation are what let the hospital say yes to our system, not just admire it in a conference talk or in a webinar. And this compliance landscape is evolving very fast. It becoming fundamentally engineering requirement, not paperwork only. So 4 pillars worth knowing as India's CDSCO, the DPDP Act, the Global ISO 42001 2023, and the EU AI Act. I will just go through briefly. So in India, we have the CDSCO, which oversees the software as a medical device. It regulates to ensure every diagnostic AI tool meets the same efficiency and safety bar as any other medical device. We have the DPDP Act of 2023, which is now active and protects the patient privacy and personal information across India's digital health ecosystem, the legal backbone that makes federated learning here both possible and lawful. We have the global ISO IC 42001, that is the new international standard for AI management system. It's how an organization proves it manages AI risk in impact on society responsibility, not just claims to. It's like we have in ISO 15189 in NABL labs. And the EU AI Act is the broader legislative signal. It regulates AI technologies to build trust and safety at a continental scale. So 4 different frameworks, one shared message. The rails of trustworthy AI are being right now in real time around the globe. Why does all this matter so much? Because the validation and regulatory frameworks are what that underpins every healthcare AI solution we will discuss in future times. So trustworthy systems will get adopted at much larger scale, whereas unregulated ones will stay stuck as impressive demos only. So let's come back to where we started. One mind, 3 major threads, beyond the chatbot into a system that is really an architecture that will accelerate research, the research that must reach everyone, and especially at the global south, and all of it held together by the governance that we can trust. That is the intelligent healthcare of ecosystem of 2030. Thank you for patiently hearing. I will stop screen sharing. So maybe we can take questions later at the end, and we can go with the next speaker. Navamita, can you introduce Dr. Sudip?
Thank you, Dr. Ray, for this enriched and insightful presentation. And your talk has clearly demonstrated that the future of healthcare lies in responsible collaboration between clinicians and intelligent technologies. So now we are moving towards the next presentation from digital pathology and intelligent diagnostics. And we now move into one of the most rapidly evolving areas of healthcare, artificial intelligence in oncology. And it is my pleasure to introduce our next distinguished speaker, Dr. Sudip Haldar. Dr. Haldar served as a consultant surgical oncologist at Shankush Cancer Hospital, Patan, Gujarat. He possesses an exceptional multidisciplinary academic background, holding an MS in general surgery, DNB in surgical oncology, a master's degree in data science, and is currently pursuing an MBA in healthcare management system. He also served as an NABH assessor and is a reviewer for JCO Global Oncology. His clinical expertise spans head and neck oncology, oral cancer, breast cancer, gastrointestinal malignancies, gynecological oncology, and urological oncology. Beyond the operating theater, Dr. Haldar actively contributes to the development of AI validation framework, clinical data infrastructure, healthcare automation, and computational decision support system, particularly for resource-limited healthcare settings. His unique perspective as both a practicing cancer surgeon and data scientist enables him to critically evaluate the opportunity and limitation of artificial intelligence in real-world oncology practice. Dr. Haldar, the dais is open for your presentation. Thank you.
Thank you for the nice introduction. Am I audible?
Yes, sir, you're finely audible.
Yes. Sharing my screen. I think my screen is visible.
Yes, sir, your screen is.
Okay, I want to start with a question. It's a real one that has been sitting at the back of my mind every time when I stand in an operating theater. I look at the biopsy report, and every time I tell a patient, we got clear margins. The question is, what if the boundary between safe and dangerous tissue is not where you think it is? Now, I'm going to ask you to hold that question for the next 15 or 20 minutes or so, because everything I show you tonight is a fragment of the answer. As already introduced, I'm trying to be in the intersection between the oncology and the data capture, data management, because the paucity of the different problems in data management and data capacity build up. And I find myself sitting at the uncomfortable intersection of what the knife can do and what the data is trying to tell us. So, in 1953, a pathologist named Slaughter described something he called field cancerization. He was looking at oral cancer specimens and he noticed something disturbing. The cancer wasn't just in the tumor, the mucosa around it, the tissue that looked normal and already abnormal. At a molecular level, the whole field was at risk. Now, 70 years, this is an important number, 70 years later, with everything we know, everything we can do, we still draw our surgical margins on anatomy. We measure 1 to 1.5 centimeter from the tumor margin so that we can get a 5 millimeter clear margin in the final histopathological report. And when the report says clear, we move on. But here is the uncomfortable truth that 30% of that oral cancer recurred at the surgical site despite clear margins. Despite our best work, we are doing everything correctly by our current standards. And another important thing is 4.6 months, like a circulating tumor DNA can detect recurrence 4.5 months before it shows up clinically. That window exists. So what is actually going on? So before that, just let me show you a patient, and 52-year-old tobacco chewer, 20 years. And he came to my OPD with a painful ulcer over the right side of the tongue. We biopsy to well-differentiated squamous cell carcinoma, confirmed. But here is what makes this patient difficult, and at the same time, it makes him completely ordinary in any OPD with oncology or head and neck oncology. He also has erythroplakia on the floor of the mouth on the other side and the leukoplakia on his soft palate. So these are potentially malignant disease. So how to tackle this thing? 3 lesions, 1 mouth, 1 visit. Now I want to ask all of you 4 questions, simple questions I face every week. Which of these lesions will transform first? The histology cannot tell me the order. Where does the dangerous field actually end? Because 5 millimeter or 1.5 centimeter, whatever the margin I take around the tongue SCC, maybe molecularly compromised tissue that the pathology reports calls normal. And Will he develop second primary? Statistically, yes. Individually, I have no idea which patient in my clinic this week or maybe this patient, I don't know. And what surveillance interval does he actually need? We give 3 months, and that is not a precision, that is a schedule. These 4 questions are what tonight is about, and by 2030, I think we will have all the answer. Now, here is why margin problem is so hard to solve. The cancer doesn't start where we see it. It starts in the epigenome, the molecular machinery of the cell, long before there is any histological changes that the microscope can pick up. It is the promoter hypermethylation, CDKN2A, T53 mutation. These are things are going on. These molecular events happen before a single abnormal cell is visible. Now, Worsham and the colleagues showed us the continuum from normal mucosa to pre-malignancy to carcinoma driven by these epigenetic changes. Ritavi and colleagues in 2025 showed us something even more important for surgeons, like loss of heterozygosity and P53 overexpression, not in the tumor, but in the histologically normal mucosa distant from the tumor. The field is cancerized far beyond what the surgeon can see. And very importantly, what Camusi's group showed that this field is not only cancerized but personalized. DNA methylation and microRNA patterns in head and neck cancer form non-random carcinogen-specific epigenetic signature for like tobacco, alcohol, or HPV as the risk factors and carcinogen, meaning that the molecular fingerprints of the field mirrors the exposure history of each patient and sets prognosis. What does this mean surgically? It means that a clear 5 millimeter margin is a histopathological concept, not a biological. It means that histologically normal tissue may carry the next cancer. It means the field boundary is invisible to every tool we currently use in operating theater. And it means that molecular map is not a supporting document, it is the surgical plan. Now here it's getting exciting because this is not a future speculation. This is already working right now. 15 machine learning studies on oral potentially malignant disorder accuracy from 74 to 95%. Random forest and unit models achieving almost 100% sensitivity. And that is the clinically usable number. Metrics is somehow difficult to interpret how to interpret this metrics. It will be altogether a different story. But if you look at the other things like Satasivam and colleagues did something elegant, like they took standard FFP biopsy tissue and extracted 11 RNA genes, RNA and 11 genes, 11, and that 11 signature identified patients with a hazard ratio of 12.65 for the worst prognosis. This is not a marginal improvement. This is a transformation predictive power. So, altogether, these tools, imaging, AI, the molecular classifiers, computational histopathology, together they form a digital twin and a living data model of the patient that updates with every clinical encounter, not a snapshot that actually we are treating like a snapshot, a siloed different parameters and different reports. So that is the architecture of what 2030 looks like. Now, I want to tell you about a study from Memorial Sloan Kettering Cancer Center published in 2026. They took an AI-driven reflectance confocal microscopy device, handheld intraoral probe, and they used it to assess the surgical margin in real time during oral cancer surgery. No frozen section, no wetting. The surgeon holds the probe against the tissue and AI reads the margin. The metrics are great. It outperforms expert. But I want to precise here, this is not the replacement for the pathologist. It is a real-time assistant for the surgeon at a moment when the pathologist is not in the room. At that moment, when I am standing at the operating table, the patient is open and I am deciding whether to take more tissue. That information is everything for me on that time. And the second tool questions every head and neck cancer surgeon face, like in every early cases, like do I do an elective node dissection? For me and other head and neck onco surgeon in India, we have some data like we have very minimum threshold to do the neck, but still is a deep learning model trained on MRI achieved an AUC of 0.928 for predicting occult metastasis and externally validated in 319 patients, but retrospectively. Now, I want to add one more data point from the ASCO 2026 because this is the regulatory signal that matters. And FDA approved Clear AI, an AI platform that interoperative based margin assessment this year. The regulatory pathway for head and neck squamous cell carcinoma equivalent is already mapped. The surgeon still decides. The AI removes the ambiguity from two hardest intraoperative question: whether is the margin and do I take the neck. Now, let me come back to our patient, 6 weeks post-surgery, come to OPD, looks well, wound has healed, examine him, everything looks fine. I tell him, see you in 3 months. Now, what if at 6 weeks, 6 week visit, I had a blood result at ctDNA level, circulating tumor DNA measurement taken from a simple blood draw. Hello and colleagues published a systematic review and meta-analysis of 3,574 patients with head and neck squamous cell carcinoma. ctDNA detected recurrence in 80% of the patients with a median lead time of 4.6 months. This is a 4.5 months of opportunity to image, to biopsy, treat, catch something before it becomes inoperable in head and neck oncology. That window can be different between a salvage surgery and palliation. So this is a blood draw, a simple assay, and this is also not only the supporting the clinical decision, this is a generating the earliest signal that drives the further treatment. So 2030 protocol for our patient is not a uniform 3-monthly schedule. It is a pre-operative field map, baseline ctDNA clearance check at week 4, updated field score at month 3, and from month 6 onward surveillance interval set by biological risk, ctDNA, not by calendar that applies equally to every patient regardless of their molecular profile. Now, same patient, same disease, 2 timelines. 2026, like today, we are doing biopsy, tongue lesions, do CT and MRI, resect to achieve 5 millimeter clear margin, and then we reported clear margin. And we watch and wait on the erythroplakia. We observe the leukoplastia. We schedule the 3 monthly OPD visit for everyone. In 2030, possibility and probably before we operate, we generate a molecular field map of entire oral cavity. We get an epigenetic risk score for every mucosal surface, like the erythroplakia on the floor of mouth scores of 0.81, so that we can excise at the same sitting. At the same time, the leukoplakia on the soft palate is with a score of 0.34. It goes on intensified surveillance. Tongue SCC is resected to the biological field boundary, not anatomical. And post-operatively, ctDNA and the field score together determine when this patient comes back and how urgently. The difference is not surgeon skill. The difference is the information available at each decision point and person who generates that information in many cases. So now, before give you the answer, so it is a live poll. If it is possible for you to scan this QR code and send me your answers, like what can't you measure but wish you could. So I can pause for 30 to 60 seconds. So just scan this QR code. It will be there in your mobile and you can type it there. If you're already in the mobile, then you can join at the slido.com and you have to give that number. I think it is somehow maybe difficult. And we can take this question later on also. Some participants are typing, so I'm just waiting for a few seconds. healthy gap from the tumor depth to potential tumor forms and likeness. So wonderful. These are the things I think we can definitely lead to the in 2030 we can achieve these things through data and other uh, tools and everything. So thank you very much for your response. Now, I owe you the honesty. Everything I have shown you is real. Studies are published. The tool exists. The evidence is solid. But I work in Gujarat in Patan. I work in a setting where the infrastructure assumption behind most of these studies do not apply automatically. Now, ASCO published a framework this year that names something I have been trying to articulate for a long time. The dominant risks in deploying AI in the healthcare are not algorithmic. These are very easy. They are socio-technical, poorly defined intended use, hidden data set, workflow mismatch, inadequate monitoring, and algorithm is often the easy part, and the hard part is everything around it. And then ASCO gives us 5 questions that every IT must answer before deployment. Is it human-centered with a clear clinical need function? Is it applicable to my core population? What guardrails exist for accurate output? Is it equitable? Right. And is there transparency with how data are used? For most published AI tools in head and neck squamous cell carcinoma, question 2 fails immediately. They are not trained on South Asian population or Indian population, or they are not validated here. And a tool that performs beautifully in a Western academic center can perform very differently in my center. But here, what I want you to leave with, because this is not a council of best practice, there are things we can build right now. You can start a longitudinal surgical registry with every patient outcome, structured data, MDTs, and also the second primary malignancy, these findings, everything. The vision is real. The path requires us to build the infrastructure it runs on, and the infrastructure starts with data capture. Now, we started tonight with a question, what if the boundary between safe and dangerous tissue is not where we think it is? Here is the answer. It isn't, and by 2030, we'll know exactly where it is. Not because of one technology, not because of one study, because of 3 shifts, anatomy to biology, margins defined by molecular field, not histological distance, from intuition to probability, every lesion getting a risk scope, surveillance personalized to the patient's biology, and siloed to integrated surgeon, pathologist, other specialists and molecular scientists working from one data layer, one shared understanding of the same patient. The AI generated the probability map. The diagnostic scientist reads the liquid biopsy, the pathologist defines the field, and the surgeon decides where to cut. And none of us replaced, all of us become more precise, and by 2030, I will be a better surgeon because of AI, not a different one. So thank you very much for your patience listening, and I'll just stop sharing my screen.
Thank you, sir. Thank you very much. Your presentation reminds us that while algorithm can assist clinicians, wisdom, empathy, and surgical judgment remain uniquely human qualities. Now we are going to continue our journey exploring the future of healthcare, and we now turn our attention to one of the most transformative frontiers in modern medicine, molecular pathology and multi-omics. The ability to decode disease at the genomic, transcriptomic, proteomic, and molecular levels is redefining how we diagnose cancer, predict treatment response, and deliver truly personalized patient care. It is my great privilege to introduce our next distinguished speaker, Dr. Indranil De. Dr. De is a consultant molecular pathologist at Netaji Subhash Chandra Bose Cancer Hospital, Kolkata, where he plays a pivotal role in advancing molecular diagnostics and precision oncology. He completed his MBBS, followed by an MD in pathology, and further pursued a postdoctoral fellowship in molecular pathology at Tata Medical Center, Kolkata, one of India's leading cancer care and research institute. With extensive expertise in molecular diagnostics, cancer genomics, and precision medicine, Dr. Dey has worked at the forefront of the translating genomic discoveries into meaningful clinical practice. His work encompasses advanced molecular technologies, including next-generation sequencing, single sequencing, real-time PCR, and digital PCR, enabling accurate diagnosis and molecular characterization of both solid tumors and hematological malignancies. Beyond diagnostic excellence, Dr. Dey is deeply engaged in translational research, striving to bridge the gap between laboratory innovation and bedside patient care. His research interests span oncogenomics, multi-omics integration, biomarker discovery, personalized cancer medicine, and the application of artificial intelligence in molecular diagnostics, areas that are shaping the future of precision healthcare. Prior to his current role, he served as an assistant professor in Department of Pathology at Dr. D.Y. Patil Medical College, Pune, where he contributed significantly to medical education and academic research. Dr. Dey has authored numerous publications in national and international peer-reviewed journals, delivered invited lectures at molecular pathology workshops and scientific conferences, and presented his research at prestigious national and international meetings. His contribution to the field have been recognized through several awards, including the highly prestigious Association for Molecular Pathology Impact Award 2023, presented in Milan, Italy, a recognition of his outstanding contribution to molecular pathology on the global stage. Today, Dr. Dey will speak on a topic that lies at the heart of next generation healthcare, that is, from the data to diagnosis, the clinical promise of multi-omics. In this session, we are going to explore how the integration of genomics, transcriptomics, proteomics, and advanced molecular technology is transforming disease diagnosis, enabling precision oncology, and paving the path toward a future where clinical decisions are increasingly guided by comprehensive molecular insight. Dr. Dey, we are delighted to have you, and now the dais is open for your lecture.
Thank you so much for the warm introduction. Am I audible to you?
Yes, sir.
Okay, I'll be sharing my screen. Is the slide visible?
Yes, sir, finally visible.
Okay, just a moment. Yeah, is the slide visible?
Not at present.
Not at present.
Just try to share the whole screen, maybe.
Okay, yeah. Yeah, I hope now it is visible.
Sir, it's not visible.
Okay.
Yeah, it's coming.
Yeah.
Yeah.
Okay. So, good evening to all of you. The topic of my talk today is from data to diagnosis, the clinical promise of multi-omics. So, this is a brief. I'm not able to share the. I'm not able to change the screen slides.
Can you share the PPT to us if possible? We can just.
Yeah, sure.
Meanwhile, if there is any questions from the participants from the first and second session, you are free to ask, so we can use the time. Any questions or any comments from any participants?
Am I able to share my
Visible?
screen now? Can you see my presentation?
Yeah, yeah.
Yeah. So then I will continue. Sorry for the technical glitch. Are the slides moving?
Yes, yes, absolutely.
Yeah. So this is a brief overview of my talk. I would like to talk to you about multi-omics and how it is being translated into diagnostics. So the central theme for our webinar today is Healthcare 2030, and it is seeing four revolutions collide. These are research, diagnostics, digital health, and artificial intelligence. And all are converging into the central theme of precision healthcare for our patients. Now, one thing I would like to make you understand is precision healthcare is fundamentally dependent on something known as biomarkers. These biomarkers are biomolecules which have diagnostic, prognostic, or predictive value. What I mean by this is a diagnostic biomarker helps to identify affected individuals from a population, while a prognostic biomarker will help to segregate high-risk patients from low-risk patients. And a predictive biomarker helps to preempt which patient will respond best to a particular therapy and which patient will not respond to a therapy. So to develop biomarkers and study them, an important thing is to understand biological information. So biological information is a complete set of molecular instructions which determines an organism's structure, function, development, or how it will respond to environmental stresses. This information can be encoded in the DNA, which gets transcribed into RNA, translated to protein, and then functional proteins come and make up something known as a metabolites. So omics is the comprehensive study of entire class of biological molecules to understand how they're affected in health and disease. So if I want to study at the level of DNA, it is known as genomics. If we are conducting a study at the level of RNA, it is transcriptomics. Proteomics is the study of proteins, while metabolomics, we are dealing with metabolites. So each omics captures a different layer. Now this particular table shows you the various omics layers with the information they analyze, the clinical application, and current testing platforms. So when I'm studying genomics, I am concerned with DNA sequences, mutations, and this is clinically applied in hereditary disease testing, pharmacogenomics, and in diagnosis and prognosis of cancer. And currently the most popular technique to study this is next generation sequencing. In transcriptomics, I'm dealing with RNA expression and transcripts. Here I want to see gene expression profiling. How are these genes getting expressed in the tissue? This is very useful in cancer subtyping and prognostication. And currently popular techniques are RNA sequencing, microarray, and PCR. In epigenomics, which involves processes like DNA methylation, histone modification, we are looking at epigenetic modifications. And these are essential to detect epigenetic biomarkers and help in cancer diagnosis and prognosis. Testing platforms available for epigenomics are ChIP-seq, bisulfite sequencing, and DNA methylation assays. In proteins, we want to look at protein expression and how proteins are interacting with each other. So this will help us identify biomarkers which are protein in nature. And also interestingly, they can help us to find drug targets which can be activated or inhibited by certain drugs. Common techniques in this are ELISA, protein arrays, and the sensitive mass spectrometry. In metabolomics, we look at metabolites and how metabolic pathways are affected. And this is very critical in metabolic disorder detection, like inborn errors of metabolism in diabetes. And mass spectrometry, NMR spectroscopy are the technologies which are used to test this. And finally, microbiomics is the study of the microbiome environment and how it affects in health and disease. So gut microbiome studies are a very interesting aspect. And also what else we have is when we are contemplating probiotic or microbiome therapy, that is where it is coming up in a great way. So metagenetic sequencing, etc., are techniques for microbiome analysis. Now, this particular slide shows us the various omics technologies which are available. There is microarrays, NGS, mass spectroscopy, microbiomics. And what I want to show you is that most of them, the output is according to data. And this data varies from few GBs to thousands of GBs. So this is something where the assay results are coming as data. So biological information has to be interpreted as data to make a sense of the information we are getting. So what is multi-omics? Multi-omics is integration of these multiple layers of biological information to give a comprehensive understanding of health and disease. So when we are studying single omics, we are following a reductionist model. We want to see the disease at its base core. In multi-omics, it follows a much more holistic molecular portrait. It gives a holistic molecular portrait of health and disease, and through that, we can get greater clinical impact and better benefits for our patients. So the question comes that why do we want to integrate? So why is not single omics good enough? So utilizing single omics, we might get an answer to what mutation is present. But using multi-omics, we can actually answer what mutation exists, which genes are active, if the proteins which are produced, are they functioning? How is the metabolism getting affected because of this? And how the microbiome is affecting this particular disease state. So in a way, multi-omics reads the entire play, where genome is the script, transcriptomes are the scenes being performed, proteomes are the actors, and metabolome the outcome. So there are different layers, but they're telling one story, and to understand that will help us make better decisions for our patients. So let me give you an example of how multi-omics can assess clinical decision making. So we can take an example of lung adenocarcinoma. Now, traditional lung adenocarcinoma, the report involves histology, stage, sometimes PDL1 expression, a limited driver mutation panel, and tumor mutation burden. So utilizing single omics or genomics, we can come to a diagnosis like EGFR mutant lung adenocarcinoma and can only predict that this tumor will respond well to anti-EGFR TKIs. However, if we analyze this particular tumor through multi-omics, we can further subclassify this into 4 subtypes. A subtype in which has a strong EGFR signaling, this will have an excellent response to TKI. A subtype 2, which might have additional MET pathway activation. Here we have to integrate MET inhibitors along with EGFR TKIs, otherwise resistance will develop. We can get a third subtype, which is immune rich, and that might be a good candidate for immunotherapy, while we can end up with a subtype which has an EMT phenotype, which will indicate a highly aggressive tumor and which will have poor prognosis. So, although all can be classified as EGFR mutant adenocarcinoma of the lung, their biology and treatment response differ substantially. So, since these assays are very expensive and data exhaustive, it is very important to carefully design our experiments to improve their repeatability and reliability. So there are 3 main factors when we want to design a study for multi-omics, we have to keep in consideration. Firstly, what is the disease characteristics? Second, what is the choice of disease model? And third is what is the sample size we want to work with, and what are the phenotypic data available. So for disease characteristics, I might want to know if there is a known pathogen for this disease, or is this associated with a genetic mutation or genomic instability, or am I dealing with a progressive disease? For the disease model, I would like to know if I would like to conduct this test on cell lines, or animal model where getting a sample will not be a problem, or in human subjects. Whenever you want to conduct in human subjects, then limitations of sample comes into the picture. And in case of sample size, I should have an adequate sample size to cater for all the confounding factors, because diet, smoking history, other things can affect my study. And phenotypic data, I will like to also integrate all the radiological findings, histopathological findings in my experiment. So once I get these experimental designing proper, then we go into studying the biomarker in each of these omics layers. And then we have to integrate this data. So there are two ways of integrating a multi-omics data. Firstly is known as vertical integration, in which we take a single sample and do multi-omics study and try to analyze it by integration. This can give me causal mapping. That is, I can find a cause. The second type of strategy is horizontal integration. In horizontal integration, we do multi-omics analysis across multiple samples in multiple time points and sometimes in multiple patient cohorts. This is useful when I want to develop a new biomarker or want to discover a novel finding. Now, what are the methods of integrating this data? The first step is known as early integration or concatenation, where we put all the omics data together into a table. The next step is to clean the data and adjust each omics data and put them together. This is known as intermediate integration. At the third stage, we do late integration, where each of the omics data are analyzed separately and then we combine the results. Now, we use network-based integration to find any hidden relationship between these omic data. And finally, we train machine learning to understand and identify patterns in this multi-omics data to make prediction. So essentially, data integration involves integration, networking, and machine learning. So this is an interesting article in which multi-omics and artificial intelligence was combined to find out biomarkers for cardiovascular disease. So the team first did individual omics study to identify biomarkers. So you can see in this table, through various different omics study, they discovered various cardiac biomarkers. After this, they utilized machine learning and integrated the biomarkers with AI using multiple tools at multiple levels. Finally, this newly studied biomarker undergoes patient testing through risk models and patient monitoring before it can finally be translated into clinical practice. So now I will come to some examples of how multi-omics studies are translating into diagnostics. And all of them centrally depend on meeting the expectations of the stakeholders. So what is the expectation of the patient? A patient wants earlier diagnosis, personalized treatment, improved survival with fewer side effects and lower cost. For the clinician, they want a greater diagnostic confidence, a precise disease classification, reliable therapeutic biomarker, and tools to monitor the disease better for progression and recurrence. And for researchers, they want to understand the disease better. They want to discover newer biomarkers, newer drug targets, and accelerate production of newer drugs into the market. But with the central theme, all these three stakeholders actually want is better patient outcome. So the first example I'm showing you how multi-omics is bridging the diagnostic gap is a study which was conducted by Deciphering Developmental Disorders Group in UK. They studied the genotypic and phenotypic data from children of severe undiagnosed developmental disorder. And two things they found out were that a single clinical feature like autism, seizure, or congenital heart disease can act many genes. This is known as genetic heterogeneity. So studying one gene at a single time will not cut it. And the second thing they found out was a single gene abnormality can contribute to multiple clinical manifestations. This is something which is known as pleiotropy. It is in oncology where the maximum use of multi-omics is currently going on, and we can consider this as a vanguard for multi-omics. So there have been multiple landmark studies in the past 2 decades integrating multi-omics with diagnostics, and they are predominantly carried out by 2 organizations, the TCGA, Cancer Genome Atlas Consortium, and the CPTAC, that is the Clinical Proteomic Tumor Analysis Consortium. And so I have listed a few of them in this table. So the first study you can see, this was the foundational study, which actually showed that multi-omics can have an impact on oncodiagnosis. And then CPTAC has conducted proteogenomic studies in breast cancer, colorectal cancer, lung adenocarcinoma, in clear cell renal cell carcinoma. And currently, they are trying to formulate a pan-cancer atlas with a pan-cancer proteogenomic classification. They're studying 14 cancers simultaneously and trying to find out hidden relationships between each cancer types. If we come to the proteogenomic landscape of breast cancer, so now proteogenomic classification of breast cancer has significantly improved the classically PAM50 subtype of breast cancer. So you must be knowing that breast cancer is currently being classified into luminal A, B, HER2 enriched, basal, and normal light. But utilizing multi-omics, we can actually classify these breast cancer cases according to the distinct pathways which are abnormal, according to their immune landscape, according to their metabolic state, and as per patient outcome. So what could classically be termed as a luminal A breast cancer on multi-omics classification can be again further subtyped into prognostically and therapeutically relevant groups. Another impact of multi-omics has been experienced in CNS tumor diagnosis, because many of you will agree that histopathology alone is very challenging to diagnose CNS tumors, and there is a chance of possibility of misclassification. But utilizing DNA methylation profiling, we can refine this diagnosis by identifying distinct molecular subgroups. So this particular study had showed that utilizing multi-omics, about 12% of cases received completely new diagnosis, while many were upgraded or downgraded according to the WHO grading, and that has inspired WHO to actually integrate molecular diagnostics in the classification of CNS tumors. Another interesting avenue which was studied through multi-omics was in the area of cancers of unknown primary. So 2 to 5% of all cancers, despite best imaging and histopathological or IHC studies, the tumor site remains undefined. But through DNA methylation signatures, we can actually accurately predict the tissue of origin. This will help us find actionable information, and now the patient can be given site-specific therapy rather than empirical chemotherapy. Another study published in ESMO stated that most of the cancers actually develop when the patient comes with symptoms. So by that time, curative treatment is very unlikely. So they used a single blood test and extracted cell-free DNA from that, and that cell-free DNA could identify more than 50 cancers if it is present in the patient, and accurately identify the tissue of origin also. Now this can be a promising strategy at the population level where screening, where actually you do not have effective screening, we can utilize this particular method. So these were a few of the oncology examples. Now, multi-omics is not confined to oncology alone. It is also an active area of study is in neurodegenerative diseases like Alzheimer's and Parkinson's disease. So this slide shows the HeLa study. It is a multi-omics study in Alzheimer's disease, including 8,000 elderly patients on longitudinal follow-up. By utilizing multi-omics, they are able to better understand the biology, give an earlier diagnosis of Alzheimer's disease, and personalize the therapy according to the patient's need. And we are moving towards precision Alzheimer's care. Another utility and active part of research is to find out the drug targets for natural compounds. Now, multi-omics can help us discover what are the targets which a natural compound activates or inactivates. And by this, newer drugs can be developed not only in oncology, but also in chronic diseases and in infectious diseases also. So this is an interesting avenue. So now I will come to what are the challenges and practical considerations when we want to implement multi-omics in diagnostics. Why is it not yet so common in clinical practice today? First thing is that multi-omics is bioinformatically very exhaustive, and there is a shortage of clinical bioinformatician and trained molecular pathologists. A skills gap exists. So for this, we need to do workforce training and utilize AI interpretation as much as possible, but with expert oversight. We should not blindly follow the AI. We have to survey it and monitor it accordingly. The second thing is all these technologies are very expensive, and the data analysis and infrastructure expenses are also quite high. And currently there is only limited reimbursement for this. So we have to demonstrate the cost effectiveness of these tests, both to the clinicians as well as to the patient, and focus on high impact clinical indications to generate revenue. The third challenge is that as I showed you, a huge amount of data is produced, and many times this data is very sensitive data. It has implications for the family, for the children, and the following generations also. So data security, consent, and what happens when we come across incidental findings is a very crucial aspect. So for this, we have to have robust informed consent, a secure data governance, and ethical oversight. And finally, there is a lack of standardization and validation of multi-omics and AI in diagnostics. But for this, we need rigorous analytical and clinical validation. And it's an aspect which Dr. Utso has also touched upon. So now I come to what are the future perspectives of multi-omics. So the future perspective of multi-omics is looking at multi-omics at the single cell level and also in the spatial multi-omics. Here, this can answer the field cancerization or molecular field theory, which Sir had just now talked about. And by this, we can actually create molecular cell atlases and give therapeutic guidance, proper therapeutic guidance to the patient. Second is we can use multi-omics in liquid biopsy and do longitudinal studies, because a patient will willingly give blood sample periodically rather than getting biopsy samples from them periodically. So that's why longitudinal multi-omics can be possible if we can harness liquid biopsy better. And this can help us to track the molecular residual disease, and if there is any recurrence, we can also understand what clonal evolution is taking place. The third aspect is that we can use all these multi-omics intelligence for pharmacogenomics and give guided prescribe according to their guidance, so that we know that which drug will work properly for a patient, which patient requires dose reduction, and which patients should not receive a drug because of expected adverse events. Finally, there is a development of a technology which is multimodal AI. Here, we are not only integrating multi-omics, but also integrating the radiological findings along with pathological findings, so that we can, and also clinical data, so that we can accurately diagnose and also discover newer biomarkers. And an interesting aspect to this is creating digital twins. Now, what is a digital twin? It is actually a virtual patient model. It's a virtual patient model which we can use to predict disease progression, treatment response, and optimize patient care. So if you see this diagram, it is showing digital twin technology in oncology. So where all the data we are generating is used to feed into a system which will create a virtual tumor model. And here we can see how simulated tumor behavior can take place, how various treatments and their optimization can affect this tumor, and get a real-time feedback and update about the tumor from this virtual model. So where does that put molecular diagnostics in 2030? Well, this will depend on a fine balance between comprehensive data generation and the practicalities of timeline, cost, and interpretive capacity. For a developing nation, we suggest a tiered approach to this. That is, for all routine cases, we can do a directed molecular profiling, where we only do targeted gene panels, IHC, FISH, and standard hematological, biochemical, and metabolite assays, and we can provide patients with a report within 7 days. But for slightly advanced case, we can go one tier up, do whole genome or whole exome sequencing, RNA sequencing, and targeted proteomic analysis, and along with the help of AI, give a report within 2 to 4 weeks. But in case of cases which are complex, undiagnosed or refractory, we will have to do full multi-omics integration along with microbiome profiling, involvement of the molecular tumor board, which will involve clinicians, researchers, bioinformaticians, and provide an AI-enabled multi-layered integrated biomarker report. But this might take 1 to 2 months. So that brings me to the take-home message for my presentation. That is, firstly, multi-omics provides a holistic systems-level view of the disease. It surpasses the limitation of studying a single biomarker alone. Artificial intelligence is a key enabler in this because it is helping to transform this huge data which is being created into clinically actionable insights. And multi-omics has already entered routine clinical practice, and it is driving diagnosis, prognostication, and personalized therapy. So I would like to say that the goal of multi-omics integration is not data maximalism. It is diagnostic clarity and therapeutic precision. The right data from the right omics layer, interpreted by the right expertise at the right moment in the patient's clinical journey. I would like to thank you for your patient audience and would like to end with a quote from Albert Einstein, which is quite appropriate for this particular field. He said that everything should be made as simple as possible, but no simpler. Thank you.
Thank you so much, sir, for your insightful discussion and your enriching ideas on multi-omics approach toward the cancer genomics and therapeutics. And as we approach at the end of the today's session, and before concluding to this scientific program, I would like to introduce several exciting educational initiatives that reflects UDGAM's commitment to building future-ready healthcare professionals. So I would like to share my screen to display. I think my screen is visible.
Yes.
So this is the introductory webinar, and it is specially designed for the students and the researcher beginning their journey in bioinformatics. And participants would like to be explore biological databases, understand primary versus curated repositories, learn about the sequence, protein structure pathway, and clinical databases, and they try to discover how these resources support omics research, disease studies, and AI-driven biomedical discovery. So this is our upcoming free webinar on biological databases, and majorly our target audience here are biotechnology, bioinformatics students from both graduation level students and master's level students, molecular biologists, genetics learner, biomedical profession. And next, I am going to share this collaborative academic initiative with ATGC Diagnostics. This is a 4-week advanced paid course on precision medicine, which integrating omics, drug discovery, clinical research for advanced level. And the main target audience would be doctors, clinicians, pathologists, and laboratory professionals, PhD scholar, researchers, life science students, bioinformatician, omics enthusiasts, and healthcare AI and drug discovery learners. And the registration is opening soon, and detail would be released very soon in our all social media platform. Next, I would like to introduce a 3-week advanced paid course that is on next generation diagnostics with Sportix Biosystem. This is a 3-week interdisciplinary course on digital pathology, microanalysis, proteomics, and computational diagnostics. And here we are going to make some digital pathology foundation, and we are going to put some flavors of artificial intelligence and computational pathology. And the target audience is pathologists, laboratory professional, researchers, biotech students, and bioinformatics learner. And registration schedule is going to be announced very soon. So I already discussed that this is only the beginning of our journey, and we invite you to stay connected with UDGAM, participate in our future webinars and courses, and collaborate on research initiative, and join us in building the future of healthcare. So together, let us transform ideas into innovation, innovation into impact, and impact into better health for all. Thank you for being part of the inaugural chapter of Healthcare 2030. I would like to invite Dr. Utso. Please deliver the vote of thanks on behalf of UDGAM AI and Health Research Center. Thank you.
So thank you, Navamita, for that introduction of our next webinars and the upcoming courses. So just a few words before we close. It's almost dinner time for everybody. So thank you to our distinguished speakers, Dr. Sudip, Haldar, and Dr. Indranil for their eloquent and very detailed talk on advanced diagnostics that are coming hopefully in the coming few years. And obviously to our participants who have been with us for the last 1.5 hours, and to Navamita for moderating the whole session. And we look forward to welcoming you all in our future events and all educational programs. Please stay connected with us in our social media pages, in our WhatsApp or our LinkedIn pages. And also we will be sharing these flyers in our all social medias, and also the registration, when the registration starts, we will share the registration links also and further details. So have a wonderful evening, and let us continue innovating intelligence for better health. Thank you all. Have a good night.